Bismillahirrahmanirrahim and Assalamu alaikum. I am Dr. Mohsin Tassawar, working as Senior Lecturer at University Institute of Medical Laboratory Technology, the University of Lahore. Today our topic of discussion is the poliovirus, and you can see the three-dimensional structure of the poliovirus in the background. As most of you know that the poliovirus is the causative agent of the well-known polio disease. So we will start from the genus of the poliovirus, that is the Enterovirus. Enteroviruses are a genus of the Picornaviridae family, which replicate mainly in the gut. These all are the single-stranded naked RNA virus and have icosahedral symmetry. And unlike rhinoviruses, they are stable in the acidic pH. The capsid of these enteroviruses is of icosahedral symmetry, which is made up of 60 copies, each of 4 proteins, and these are VP1, VP2, VP3, and VP4. And most of these viruses contain positive sense single-stranded RNA strands. At least 71 serotypes are known, which are divided into the 5 groups. One of them is polioviruses. Second, Kawasaki A virus, Kawasaki B virus, ecoviruses, and enteroviruses. Here you can see the electron micrograph of these enteroviruses, and you can notice the icosahedral symmetry of these viruses in this electron micrograph. Talking about the history of the poliovirus was firstly identified in 909 by inoculation of the specimen to the animals like monkeys, and the virus was first grown in the cell culture in 1949. That was the basic foundation of the cell culture based vaccines. Talking about the poliovirus have 3 serotypes, which are designated as poliovirus serotype 1, 2, and 3. But these all 3 serotypes don't share any antigen. They have identical physical properties, but they only share 36 to 52% nucleotide homology. Another thing is humans are the only susceptible host for these viruses. It means that they cannot replicate in other species like birds or some other animals. Definition of polio disease can be done as polio is a contagious viral illness that in its most severe form causes paralysis, difficulty breathing, and sometimes death. The other names of polio is poliomyelitis, infantile polio, and the polio disease. Talking about the transmission of the virus, there are 2 major modes of transmission of this virus. First one is fecal-oral transmission. Another is oral transmission. The fecal-oral transmission is done due to the drinking water or some contaminated food. And most commonly, in this case, the virus is present in the sewage water or the infected person's feces. In the oral transmission, when the infected person sneezes or coughs, so it can disseminate the virus into his surroundings in air. Talking about the structure of the virus, it is somewhat simple structure of the virus because it is a naked RNA virus, so it only contains capsid and genome. The capsid of the poliovirus surrounds, delivers, and protects the RNA. That is the major function of the capsid in every virus. The capsid of poliovirus is icosahedral in its structure, which has 20 equilateral triangular faces. The capsid of poliovirus further consists of 4 different proteins, as we have discussed earlier that all the enteroviruses are made up of 4 different proteins, VP1, VP2, VP3, VP4. So no difference in the case of polio. The VP1, VP2 and VP3 particles are present on the surface of the poliovirus, while the VP4 particle is present inside the capsid. Genome, as we have discussed earlier, that poliovirus contains a positive sense single-stranded RNA genome with the length of 7,500 nucleotides. Receptors, that is a very important point, because most of you know that every virus attach with the specific receptors. So in the case of poliovirus attaches on the CD155 receptor site. These receptor sites are also known as poliovirus receptor sites, or some other places you can say that is immunoglobulin like domains. And the virus only bind to that cell which contains these receptor sites. So that is the same thing in the case of all viruses. Talking about the distribution of the poliovirus is present globally everywhere in the world, especially in the developing countries. And before the availability of the vaccines, almost 100% of the population under the age of 5 were infected with the polioviruses. With the availability of immunization, and the poliovirus eradication campaign has eradicated poliovirus in most regions of the world except in the Indian subcontinent and the Africa. Poliovirus is still endemic in 3 countries, which include Afghanistan, Pakistan, and Nigeria. All other countries have eradicated the poliovirus. WHO actually set the target of year 2000, by which they plan to eradicate the poliovirus globally, but still in these 3 countries you can find the endemic of this poliovirus. While talking about the pathogenesis of the poliovirus, the incubation period varies between 7 to 14 days. Following the ingestion, the virus multiplies in the oropharyngeal and intestinal mucosa. The lymphatic system, particular tonsils, and the pear patches of the ileum are invaded, and the virus enters the blood, resulting in a transient viremia. That is something very important in a viremic condition. In a minority of cases, the virus may involve the CNS following the dissemination. After involving the CNS, it may cause the paralysis. Talking about the clinical manifestation of the poliovirus, there are 3 possible outcomes of the infection. First one is subclinical infection. That is 90 to 95%. That is inapparent subclinical infection account for the vast majority of poliovirus infection. That means 90 to 95% of the infected people, the poliovirus remains asymptomatic. They do not show any symptom of the virus. Second outcome is abortive infection, that is present in 4 to 8% of infected people. That is a minor influenza-like illness occurs. Recovery occurs within a few days, and the diagnosis can only be made by the laboratory. So these are the minor influenza-like illness. That is an illness that is commonly present in almost every viral infection. In this case, you cannot diagnose the poliovirus with the symptoms. You have to go to laboratory for the confirmation of the virus. The minor illness may be accompanied by aseptic meningitis. Third outcome is major illness, that is paralytic and non-paralytic poliomyelitis, that is present in 1 to 2% of the infected people. The major illness may present 2 to 3 days following the minor illness or without any proceeding of the minor illness. Signs of aseptic meningitis are common. Involvement of the anterior horn cell lead to flaccid paralysis. And involvement of medulla may lead to respiratory paralysis, as you know that medulla is the part of brain which is responsible for the respiratory system and the heartbeat. So in the case of involvement of medulla, the patient may feel the respiratory paralysis. In simple word, abortive poliomyelitis is the mildest form. Non-paralytic poliomyelitis symptoms are more severe than abortive, but not as bad as paralytic. In paralytic poliomyelitis, the most severe may result in permanent paralysis of certain muscle group, as we have discussed earlier, like breathing muscles and leg muscles. The laboratory diagnosis of the poliovirus, the first thing is you can isolate the poliovirus from the throat swab, feces and rectal swab, but it is rarely isolated from the CSF. The poliovirus can grow readily in the cell culture and can be identified in cell culture using different CPEs. So you need to require the molecular methods to differentiate between the wild type and the vaccine type of poliovirus. The very rapid method to diagnose the poliovirus is RT-PCR, that is reverse transcriptase PCR, which you are going to use in the case of every RNA virus. So that is very rapid method to detect the poliovirus. Another method is serological method, but this method is used very occasionally because that is not so efficient as cell culture or the RT-PCR. So these serological methods are used for the screening of massive individuals or mass population. Talking about the prevention of the poliovirus, there is no antiviral therapy available for this virus. However, the disease may be prevented through the vaccine. There are two types of vaccines available for the poliovirus. First one is IPV, that is intramuscular poliovirus vaccine. That consists of formalin inactivated virus of all three poliovirus serotype. If you remember, we talked about the three different serotypes of poliovirus. So these IPV vaccines contains all three serotypes of the poliovirus, so that they can provide immunity against all these three serotypes. The IPV vaccines produced serum antibody only. They do not induce the local immunity like in the case of IPV. The immunoglobulin IgA will not be produced, so there is no local immunity present in the case of IPV, but there would be a lot of serum antibodies, protective antibodies. However, it will prevent paralytic poliomyelitis since viremia is essential for the pathogenesis of the disease. So in simple words, if you are using IPV, maybe in some cases a patient can get a minor symptoms of the polio, but viremia will not be developed, so there will be no chance of the paralytic poliomyelitis. And other vaccine is oral poliovirus vaccine, that is OPV, and it consists of live attenuated virus of all three serotypes, and it also produces local immunity like IgA and rarely cause paralytic poliomyelitis, around 1 in 3 million doses. That is very much high success rate of this vaccine. Most countries use OPV because of its ability to induce local immunity, and also it is much cheaper to produce than IPV. And the normal response rate to OPV is very close to the 100%, like it is around 100%, very much efficient vaccine. OPV is also used for the WHO poliovirus eradication campaigns. Thank you very much. It was all about the poliovirus and the vaccines available of the poliovirus. Thank you.